All right, so we need to get back into the light reactions and think about that whole process when we were, where is the, okay, I'm going to share the screen. All right, hopefully just one big slide. Okay, so we're looking at the light reactions. So last week we ended up talking about pigments and photoreceptors, and that there are many different kinds of pigments depending on the organism that's doing photosynthesis. And then we have the idea of, let's think about leaves, the typical larger plants, and we're going to think about the leaf and all the different cells inside the leaf with the chloroplast. So when you look at the anatomy of a leaf and look at its internal design, it's really exquisitely designed to capture light energy. And it also not only capturing light energy, but then funneling it through the tissue to all the different cells. And so the design of the leaf is really well adapted to maximize the efficiency of sunlight absorption. So one of the things that plants routinely do through the day, if you're thinking about a plant out in its native habitat where it has to deal with the moving sun across the sky, is it always tries to what's the word? I'm blanking on the word. It always tries to get its leaf blade in orientation, there we go, in the orientation 90 degrees to sunlight. And so as the sun comes up, you will even see certain plants through the night will start to shift their leaves so that their leaf blades are 90 degrees to the, when sunrise hits in the east. And so plants have this ability to really, right from the get-go, capture sunlight energy right at sunrise, keep their leaf blade at 90 degrees orientation to the incoming sunlight beaming down on their leaves. And then they'll track the sun across the sky most typically. So, the dramatic example that they often give for that is a field of sunflowers. You will often see the actual sunflower, the whole plant body turns and orients to the sun. Then, so not only can it manipulate the orientation of its leaves and change its direction, so to speak, if you look into the leaf tissue, the optical properties in there, so the arrangement of the palisade mesophyll cells and the arrangement of the spongy mesophyll cells, and then all the empty airspace in the tissue is designed in such a way so that as light is coming through, certainly the palisade the palisade cells, they're stacked tightly together and kind of column-like, but they will allow light to pass through down into the spongy mesophyll area, and then that's where the open airspace is when you look at the tissue, and then the light refracts off of that area and bounces around, so that pretty much all the cells inside the tissue, the leaf tissue, are getting hit with light energy. And then of course, if you look at the vein pattern in a leaf, it's highly vascularized, which means that once the plant has done photosynthesis and made all those lovely sugars, it's able to get it into the xylem and while the phloem first transport it. Tiny, right? Yeah. So the question was about, what about gymnosperms and conifers where you have the needle-like leaf, and they don't really orient their leaves so much, and so yeah, their design is very different than the typical oak leaf, maple leaf, and actually, to be honest, there's not a lot of research that I'm aware of that's being done on the efficiency of light capture with needle-like leaf design. The plant physiologists seem to focus so much more on the angiosperms. So I don't have an easy answer on that one. I mean, they're certainly doing photosynthesis, and the nice thing about gymnosperms in around here for us to enjoy is, you know, they stay evergreen through the winter. And so even on winter days, they're capable of doing photosynthesis if it's warm enough. And they can kind of activate for brief periods during the winter season. But not a lot of research done on their dynamics of how they do light capture and photosynthesis in any detail. So it's quite a bit lopsided. The angiosperms get the lion's share of all the research. Each cell inside the leaf typically has many chloroplasts. So an average range that's thrown out a lot is 20 to 60 chloroplasts per cell. So, and then guard cells, it depends on the plant species. Some plant species have chloroplasts in their guard cells, others do not. I would say most, probably greater than 50% do, but not all. And so we're going to be zooming into the chloroplasts, of course, because that's where both the light reactions and the biochemical reactions are taking place. So this is just a refresher to get the right neurons firing. The chloroplast is bounded by that double membrane. So there's an outer membrane and inner membrane. The inner membrane is a little bit more selective than the outer membrane. And then, but what we want to do is really go further than that. We want to look at the internal membrane, which is the thylakoid that's floating around in the inside the chloroplast. So the thylakoid membrane, if you were to take a tiny little scalpel and cut into the thylakoid membrane, you would see the internal portion of the membrane, and that's called the lumen. Just like our intestinal tract, when we talk about where the food is moving through, we call that the lumen portion of the intestinal tract. So it's just that opening bounded by, whether it be an intestine, tissue, or the thylakoid membrane. And so the lumen, I do need you to know that because we're going to talk about certain things happening in the lumen, certain other things happening in the stroma, and then all these terms. So lumen, grana is the stacked regions of the thylakoid. You need to know that, please. And the stroma lamellae, again, is the relatively unstacked region. So this is from probably week 1 material. I'm just trying to get you back on track on thinking about the chloroplast. And so it's the thylakoid membrane that we're zooming into for the idea of that's where the chlorophyll is embedded. The phytol tail of the chlorophyll pigment is embedded in the thylakoid membrane, and that's where we have the absorption of light energy and the light reactions. So we have the pigments and then the proteins and enzymes and other factors all combining in the thylakoid membrane. And then the stroma portion inside the chloroplast, when you crack open the chloroplast and look inside, we call that region the stroma. And that's where we're going to do part 2 of photosynthesis, where they actually do the biochemical reactions to take the CO2 and make sugars. Okay, so we're zooming into the chloroplasts. Again, you've seen this picture before. So that internal membrane, and you can see there that there's relatively stacked and unstacked regions, the grana and the stroma lamellae. And here's the drawing of it. And then if you take and cut into it and peer inside the thylakoid membrane, the inner portion is called the lumen. So we need to think about that because certain things are happening in the lumen and then certain things will happen in the stroma. Okay. So those pigments, the pigments are absolutely necessary. We got to get that absorption of light energy. And so we talked about the photoreceptors last week, how there's chlorophyll A and B, primarily in the higher plants, and then we have carotenoids, and then there's flavonoids, things like this. All right, so these pigments, they actually do call them the antenna complexes, kind of like the old-fashioned, rabbit ear antennas that you have on your TV sets. That the job of the antenna on your TV is to take the radio signal and get it into your TV set. Well, it's the same idea here. These pigments form antenna complexes where all these pigments are gathering light energy, and then they funnel that energy, so to speak, to a very special chlorophyll pigment in the reaction center. So we're going to talk about reaction centers, and then there's pigments that surround the reaction center and help absorb light energy and then deliver it to where it's needed in the reaction center. So it's a set of pigments, these antenna complexes. Their job is to gather light energy, and because they're slightly different pigments, like the carotenoids absorb different wavelengths, maximally, preferentially absorb different wavelengths compared to chlorophyll A and B, for example. And I'm saying that they transfer energy, not electrons. All right, so it's an energy transfer, that tuning fork analogy. So that means we're getting into that inductive resonance idea, where we have the energy transferring from one pigment to the next, to the next, to the next, until finally that energy reaches the reaction center where a very special chlorophyll is located.
I don't understand.
Madeline, do you have a question?
Yeah, can you hear me okay?
A little bit, if you could speak up just a little.
Is this better?
Yeah.
Okay. I don't understand.
Oh, you figured it out again. You're fading out.
I'll put it in the chat.
Okay. And so let me show you a picture of this idea, kind of a mock-up of the concept. Is it in the chat? Okay, go ahead.
Do they transfer the energy via like a confirmation change, or
Yeah, there's got to be some level of shift going on at a confirmation, at the physical level to get that vibration energy to transfer, essentially. So, it's a very efficient way and fast way of getting that energy going from one pigment to the next. And basically, it creates it's a very quick way to get that energy down into the reaction center. And so it makes sense when they did all the thermodynamics and they looked at the kinetics of all of this going on. It makes sense that each reaction center has this like dedicated group of antenna pigments that can all be busy funneling that energy to the reaction center. So that's what you're seeing here in this picture. They're trying to show, so P680 is the reaction center. That's the name they give one of the reaction centers. And then they mapped it on this energy figure where, higher energy, the further up you go on this graph. And then the bottom line is your unexcited state. And so they're saying when those pigments absorb light energy, it boosts them to a higher excited state, and you get this energy transfer with some heat loss. It's not 100%. And then you finally get that energy into the reaction center. So here's another image of it, another drawing. And I like this one because they're very clear to state, okay, this is energy transfer with inductive resonance, where you have that energy, with these physically closely associated pigments interacting, you get that energy transfer. And then when you get into the reaction center, they're finally showing now you see an electron being donated off. So it's two different concepts here. Very efficient way to set it up so that you have multiple pigments all feeding that light energy into the reaction center to keep the reaction center going. And these are picosecond reactions, and they're very, very quick. I mean, just one after the other. If everything's going okay, that reaction center is very busy all the time when the sunlight is hitting the plant. Where's the chat? Chat. Well, I see the chat on mine now. Sometimes I don't understand what inductive resonance is. It's not chemical, so don't think about a chemical reaction. It's the radiant energy. There's no electron transfer between those antenna pigments at all. It's just this energy transfer. And so I don't know, for me, the tuning fork analogy just totally works for me, but
it makes sense, like the analogy, but what is the intention?
So yeah, let me see if I can find another way of presenting it besides the tuning fork. Maybe somebody else. Let me look into that, because I know the first time I heard it too, it was hard to wrap my head around it until someone finally told me the tuning fork analogy, and I'm like, oh, that makes so much more sense. But
No, I get the analogy. I don't like that's not. It doesn't explain what's happening. You know what I mean? Like, are they like? Is it kinetic energy? Is it like?
well, the sunlight is electromagnetic radiation.
Right.
So that absorption of the light energy, I guess I think of it kind of like this vibrational energy transfer is how I like to think of it. Like when you get a tuning fork humming, when you strike a tuning fork and get it going, you can actually feel the vibration in your hand. And so I kind of imagine that kind of energy transfer where there's that excitation transfer of energy without any electrons being donated or accepted off. So let me
So
see.
would it be like a transfer of like radiation, like it's being like re-emitted? Like, I don't know. I guess, yeah, I guess that's my question.
I can, the chat will save at the end here and I can go back and I will answer any questions that I can't answer directly during lecture. I'll try to track a different analogy down and see if that will help. Because yeah, it's a weird thing to think about, especially the first time you're presented with it. So, this whole thing, we talk about it now pretty matter-of-factly, but it took a long time to figure this all out. Of course, they started back in the, 1600s, 1700s, and it took a long time. So this was not something quick to be realized. It was, back in the 1700s, a group of people led by Dr. Priestley, he's the one who said, yep, oxygen's definitely given off during the light reactions of photosynthesis. And then they came along and said, you absolutely have to have light for oxygen evolution. They're linked. So I'm just wanting to get across the point that this was a very long process, very methodical, and limited, of course, by the technology at the current time of the research. But let's fast forward a couple hundred years. And the top bullet is usually how you get presented with photosynthesis. This equation is the big picture, what I call the big picture idea of photosynthesis. It's covering both what's happening with the light reactions and the carbon fixation reactions, because you see the CO2 coming in, water, and then C6H12O6 plus oxygen. Now, I think I might have asked you at one point already, when I say C6H12O6, what sugar do you think of? Glucose, 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 of course, and that's what I think of too. So in this class, I don't want you to know the top bullet point. I want you to know the bottom, which if you're not colorblind, it's in red. So the second bullet point is the one I prefer you. If I ask you to write out the equation, that's what I want, because I'm trying to get you to think about the idea that it's not glucose as the first sugar. And so we represent a basic carbohydrate building block as CH2O with parentheses around it. And that's more appropriate, I think, for this level class, that you understand that, yes, we're building carbohydrates. It's not glucose first off the bat here. Okay, so let's simplify it down that way. So the other thing that should be somehow represented in this equation and never seems to be is the idea that light. So usually when I'm writing it out, above the arrow, I put, light, and then below the arrow, I put chlorophyll. And you gotta have that light absorption by the chlorophyll pigment. And so obviously that's, you'll know that for sure by the time we're done with all this. Then part 2 of photosynthesis is the carbon fixation. So when we say fixation, that word, they mean that it's getting put into a pathway that makes a more complicated molecule from it. And so a sugar in this case. So CO2 is getting fixed, but if you want to be more chemically minded about it, you would say it's getting reduced to become a carbohydrate. But a lot of people say carbon fixation. That means you're just building, you're taking CO2 and building a more complex, in this case, sugar. And then we say that water is the ultimate electron donor during the light reactions, and you'll need to know that. And that when you look at the big picture equation, CO2 is going to be accepting electrons to get reduced to a sugar. So these are some of the more bigger picture ideas, and now we're gonna really zoom in to just the light reactions. We're gonna leave part 2, the carbon fixation, the biochemical reactions to make sugar. We'll wait on that and talk about that later. So if you look at the light reactions and you look at the whole thing in total and you say, why did this evolve? Why do plants do it? What is the payoff? This is it, okay? Why are they doing it? To generate these 2 molecules, NADPH and ATP. Why? Because you have to have those in order to fully make sugars from CO2 that the plant will take in. If you don't have enough NADPH and ATP, you will not have very good sugar production. Okay, so when you look at the grand big picture idea of the light reactions, this is the payoff, which is very good. And then it's getting made, you know, all of this is happening in the chloroplast. So it's physically made right where phase 2 is going to happen with the carbon fixation. So once NADPH and ATP is made, it doesn't have to get transported anywhere. It just gets immediately used in the stroma for carbon fixation. Okay. So now to talk about the light reactions, we need to think about redox reactions. And so we need to think about the payoff, the Gibbs free energy process here, which is the energy available to do work. Now this is going back to, I think, Chem 112, maybe Chem 111, I can't remember which one, but looking at the energy available to do work. And then if, you know, if you're, I'm just trying to jog the right part of your brain here. Enthalpy, you probably learned about that at the same time. The delta H, looking at total energy, heat and work energy. T is your temperature, and S is entropy. So if you do calculations, which we're not going to do, but if you were to do calculations on different biochemical reactions, the free energy can either be a positive value or a negative value, depending on which reaction you're looking at. So if it's a positive value, that means you're going to have to push that reaction with an input of energy. Fancy word is endergonic. I don't really say that, but you might have learned it that way, so I put it down in case that's how you learned it in chem classes. If the sign is negative, if your delta G is negative, that means the reaction will happen spontaneously. It does not need any kind of input of energy. If you put those molecules together, they're going to react as that equation, that chemical equation is stating. So the fancy word for that is exergonic. All right, so let's go back to the equation and think about this now. So that big picture equation, if you take CO2 and you say, let's create some chemical equations here to really look at how CO2 is reduced into sugar, and you run the thermodynamics of that and you calculate the free energy, your delta G is going to be positive. So to take CO2 and fix it into a carbohydrate, you got to push that with energy and reducing molecules. So it's a positive delta G. I mean, that's one reason you can't go out on a rainy day like today and stick your tongue out and get sugar falling down from the sky, right? We don't have that. We have water in, you know, water vapor, and now we have rain out there. We have CO2 certainly in the atmosphere, but unfortunately we can't just go get a sugar fix by, you know, sticking our tongue out outside. So obviously we can't, even though you put CO2 and water together, you're not going to get any kind of sugar formed unless you push it with some energy and reducing molecules. Oxidation of glucose, if you flip that equation, so that second bullet, you know, let's just flip it so the carbohydrate is the substrate, and you strip electrons off of it, well, that has a negative delta G. That's essentially glycolysis. And you have and respiration all put together, I guess. And so you have, that will just happen. Okay, so when we're looking at the light reactions, we're seeing this process of electron transfer once we get into the reaction center. So we're looking at redox reactions, to shorten that down. So if you haven't thought about this for a while, I'm going to try to jog your memory. So this is the actual process where electrons are transferred from a donor molecule to an acceptor molecule. Now, when I first learned this, and I think it's still true today for the most part, very often you'll get presented with an equation that's hard to kind of think about electrons. You look at it and you go like, well, what's going on with the electrons here? So you might get presented with something like this. AH2 is the substrate, and then the chemical reaction is it breaks down to the A molecule plus 2 electrons plus 2 protons. So H2, I want you to zoom in on H2. Hydrogen is composed, each hydrogen atom is a proton and an electron, right? So when you think about this idea, it's often when you look at a chemical equation, what they show is a transfer of hydrogen during that reaction, and they'll say that's an electron transfer. But you have to remember a hydrogen atom does have an electron. Each hydrogen has an electron and a proton. And so as you have this reaction happening where the hydrogen is getting transferred to the B molecule, it really is a transfer of electrons. And I don't know, for me, when someone finally explained it that way to me, it clicked in my brain. So if that helps, fine. And so we're looking at this idea where often they'll say, oh yeah, look at this redox reaction. And it's really, you're seeing hydrogen getting transferred, but there's an electron with that. All right, so if you can keep that in mind, I think this will make a little bit more sense as you work through it. All right, now when we look at the light reactions, we're going to see that there are some players in the game that are what we call a strong reductant. That means, chemically speaking, I like to say it's very happy to give off electrons. It's just very well suited, chemically speaking, to doing that. So an example of that when we get into the light reactions is this protein called ferredoxin. We abbreviate it down to FD. By the way, all the abbreviations I use, that you can abbreviate that way on the next essay exam. All right, you can do that. Wow, we got a lot of chats going on. Okay, anything I need to be aware of right now? All right, thank you. So ferredoxin, it's very happy, chemically speaking, to give up electrons and donate it to some other molecule nearby. If something is called a strong oxidant, that means it's very happy, chemically speaking, to accept electrons. All right, so if you are a strong oxidant, that means you really you do not like to donate electrons, but you're very good at accepting. So it's kind of an either or idea here. An example of a strong oxidant would be water. So what we're saying here is water, when you look at its chemical nature, it does not want to give up electrons. Absolutely does not want to. And so you have to force the situation, so to speak, by putting in a burst of energy, which is what the light absorption, that part of that process, that's what we have happening. So let's look at it visually. Sometimes you see this in chemistry classes where they have the electrons represented in their ground state. So they even colored it green to get you thinking about chlorophyll. So in the reaction center, you have these special chlorophyll pigments there. And when that light energy is getting funneled into the reaction center from the antenna complex pigments, it's going to excite that chlorophyll and boost that electron up to a higher excitation state, higher energy state. And so now, once it's absorbed that light energy, now that chlorophyll pigment is a good reducing agent. It's ready, chemically speaking now, to give off an electron to a donor. But you have to hit it with light, otherwise it's not going to want to do it. And same with water. We have to really force the situation with water, and we'll get into that. So often you see the light reactions represented like this. They call it the Z scheme because if you tilt your head, it looks like a Z. What they're doing here is putting it on an energy scale and saying that you have when we get down to it, we're going to see two photosystems. P680 is the name of one of them, and P700 is the other. Those are the reaction center names. And then the entire thing is called photosystem 2 and photosystem 1. And then these green, they're trying to show those antenna pigments. So they're showing this absorption of light. P680, by the way, 680 is the nanometer wavelength that it really likes to absorb, which is red light. So that's why you're seeing that. And 700 is slightly beyond that. It's far red light. So you see this electron getting boosted up the energy scale here in this figure. And now you see this downhill slide, so to speak, of electron transfer. So you get it excited by light energy, and then it gets donated, you know, donor to acceptor, donor to acceptor. But you see there's a drop in energy. But you still have the opportunity. There's enough energy here to do something. So we're going to talk about that. So it's not just electron flow we're concerned about. There's something else going on here that is important, and we'll get into that. All right, and then you see both photosystems need to get hit with light energy, and that has to happen. You have to have this coordinated effort where these two photosystems have to be in concert with each other and working through the light reactions in a similar way, so you get this constant, even flow of electrons between the two photosystems. And you can see that when the photosystem 1 gets hit with light energy, it too has a chlorophyll in its reaction center, and that chlorophyll electron gets excited.
Both of those have to be present in order for it to work properly. Do they get that like whole name, like a complex name or something?
No, in fact, it's really kind of weird. So the question was, do both photosystems, you know, they're working in concert with each other. Do they have to be near each other and really coordinated that way? And actually, not really. It's kind of weird. And so I'm going to show you a picture of how they're embedded in the thylakoid membrane, and it's bizarre. So we don't give it one big name. So for ease of talking about it and explaining it, I'm going to go in a very orderly way. I'm going to talk about, let's start at photosystem 2 and let's see what happens there. And then we're going to work our way all the way to photosystem 1. But it's not so neat and tidy in the actual thylakoid membrane. And I'll show you a picture of that in a minute. It's kind of bizarre. Okay, so here is a little bit more detail of the Z scheme. And again, now it's showing a little bit more of the players in the game, where you have these donors and acceptors of the electron transfer process between photosystem 2 and photosystem 1. And so here also you can see in the light reactions, water is actually part of photosystem 2. So when water gets split, when we strip electrons off of water using that light energy, it's happening in photosystem 2. And so when you strip enough electrons off of water, you'll get the evolution of oxygen. And of course, one of the other products will be protons, because if you strip electrons off, you're left with protons off of hydrogen. So that's why we say water is the ultimate electron donor. Water is the source of, yes, we have a chlorophyll pigment electron getting excited, but once that chlorophyll loses an electron, well, it can't keep doing that. Otherwise, it's going to run out of electrons. And so it needs a source of electrons to fill the hole by the missing electron that just got boosted off. So water plays that role. The electrons coming from water will replace and go to the chlorophyll molecule and replace that electron that was just boosted off to an acceptor. Happens in picosecond timing. This is very, very quick. Very, very difficult. So the oxygen evolving complex is part of photosystem 2, and they don't still know fully how it works. It's still kind of mysterious, and it's a pretty amazing process because, like I said, water does not want to give up electrons. And so this is a pretty profound process. Then you see P700, so that's photosystem 1 getting hit with light energy again, boosting that chlorophyll electron. And so you have to have both photosystems getting hit with light energy to keep this going. Otherwise, it'll grind to a halt, and the electron flow will stop at some point in the process here, and that wouldn't be good. Then you see at the very end here, you see NADP plus going to NADPH. So we say that NADP plus is the final electron, the ultimate electron acceptor or final electron acceptor. That's the end. So here you're seeing NADPH getting made through the electron transfer chain process, which is great. The plant needs that. The other thing that's not drawn on here is the other chemical I said that's part of the big picture goal of the light reactions, which was ATP. All right, so we still have to talk about how ATP is getting made. Here's another view trying to show, again, water as the ultimate electron acceptor. You see the electron coming from water to replace the electron that was lost from that excited chlorophyll. That's getting boosted to a higher energy state, and then that chlorophyll electron gets sent off, then the water electron takes its place. And you have that happening over and over in picosecond timing. And then it goes on down the electron transport chain. Photosystem 1, you also have to have light energy striking that to keep the two photosystems working in concert. It's kind of like a marching band. You have to have everyone in step or things look really chaotic and it just doesn't work. But if you have all that happening in step, then you can have this electron flow ultimately starting from water and ending up at NADP+. Okay, so we have two photosystems to think about. They named them in order of discovery. So photosystem 1 was discovered first. But when we describe the light reactions, we usually start with what's happening at photosystem 2, because that's where water is getting split. So we like to think of it that way. Both of these photosystems, they have to work together. They work in concert. They operate in a series or a chain. And basically what we're seeing when you look at the big picture idea here is light energy is getting used to strip electrons, extract low energy electrons from water. Water hates to give up electrons, chemically speaking, and then get that excitation, raise it up to an energy level that will allow for the production of NADPH. NADPH is a strong reductant. That means chemically speaking, it's very happy to give electrons to somebody else, some other molecule. All right, so once NADPH is made, then it can participate in another series of reactions to make sugars from that, from the CO2 coming in. And it's very capable of pushing key steps of the carbon fixation by donating off electrons at key steps. All right, so now let's kind of zoom in to the photosystem. What are we talking about? In general, and they're still studying the light reactions, especially the oxygen evolving, but they're still looking at the photosystems in more detail. But what they do know so far is that there are several different proteins, chlorophyll for sure, chlorophyll A, and carotenoid pigments are there too. And then we consider the antenna pigments, they're right there, right by the reaction center. So they're, just kind of part of the photosystem. And then when you zoom in to the so-called reaction center, and so it's just that when we say the reaction center, we're zooming into where the electron from a chlorophyll pigment is getting lost, actually donated off. So that's where the photochemical reactions taking place. So at the reaction center, it's in the photosystem, and there's a cluster of chlorophyll molecules. And then one of those chlorophyll molecules, its electron gets boosted to an excited state to the point where it can get donated off. And then there's some other proteins that are complexed with it. And so it's the actual reaction center of the photosystem. That's where you actually see the photochemical reaction. And when we say that, we mean the electron from chlorophyll is given off to an acceptor molecule. So that's the chemical reaction going on. And you have to have light energy to push that. Otherwise, it's just not going to happen. So when you crack open a chloroplast and you start examining the thylakoid membrane and you look at, okay, where are all the players of the game situated in the thylakoid membrane, you see it's not so neat and tidy as how we're going to display it and talk about it. So there's certainly, of course, photosystem 1 and photosystem 2. Then there's another big complex that we call cytochrome B6F. And then there's another big complex, the ATP synthase, to get your ATP made. So these three bullets, these are complexes that are quite large, embedded in the thylakoid membrane, and they're so big, they don't move very well in the membrane. They have limited mobility because they're just so big. So remember, in membranes, you can have things moving laterally through the membrane, you know, proteins and molecules. You can have that happening. But these are so big, it's difficult. So we call them integral membrane proteins because they span the entire width of the thylakoid. They're big. Some people call them transmembrane. And then this isn't a big deal, but people will say that they're arranged in a very particular way, and they had to give you yet another term to memorize. So they call it a vectorial arrangement. Basically, what that means is that these complexes are arranged, they span the thylakoid membrane in such a way that it creates certain things to happen on one side of the membrane and certain other things to happen on the other side of the membrane linked to that complex. And you'll see this, you'll figure this out as we work through this. So it's just really well designed to allow for everything to happen to get your NADPH and your ATP. So what we see when we have this very special way in which these big complexes are inserted into the thylakoid membrane, when you look at it, it allows for water to be stripped of electrons on one side of the thylakoid membrane and NADPH to get reduced on the other. And that's important because that allows for ATP to get made. And that may make no sense at the moment, but I will show you a picture to get that point across. So water is getting stripped of electrons, it's getting oxidized in the lumen of the thylakoid. So the inner part of the thylakoid membrane, water is getting stripped of electrons. Well, if you take water and strip electrons off of it, you're left with protons. So what this ultimately means is that in the lumen, we're seeing a buildup of protons. That's important. We'll talk about why. On the other hand, the very important NADPH and ATP, they're getting synthesized in the stroma, which is on the other side of the thylakoid membrane. It's not the internal part of the thylakoid membrane. It's the part that's bathing the thylakoid, the outside part of the thylakoid membrane, so to speak. And basically, this is important with water being stripped of electrons in the lumen. It creates a really nice situation where it allows for pretty efficient synthesis of ATP. And you'll get that in short order here when we look at it and get into it. Okay. Now, if we look at all these players in the light reactions and say, where are they in the thylakoid in general? Because we talked about, you know, there's the grana region where you have stacked membranous material, then there's the stroma lamella. Well, it's weird. So photosystem 2, when they have actually analyzed this and looked at the structure, they see photosystem 2 tends to be more abundant in the stacked region of the thylakoid membrane. On the other hand, photosystem 1 and the big ATP synthase complex, they tend to congregate more in the stroma lamellae region. And then cytochrome B6F, well, that one seems pretty even Steven. They see it pretty much through the both areas, grana, stroma lamellae, it's there. So that led them to think about, okay, since we don't have them neatly lined up right next to each other, and they're such big complexes where they have limited mobility, there must be other players in the game that can shuttle electrons around and are mobile enough to flow between the photosystems and do their job of transferring electrons from donor to acceptor. And so that's what we see. We see these mobile carriers, so they call them mobile electron carriers because they are actually mobile in the thylakoid membrane. They can physically move laterally associated with the membrane in some manner so that they can get electrons from one player and then physically shift and deliver it to the next player in the game. So let's look at a picture of that where they're trying to capture that idea. So here you see most of the image is the Grana region, and you can see that photosystem 2 is the kind of the olive green. And then you have light harvesting complex 2. Those are just the antenna pigments that are closely associated. And then photosystem 1 is kind of the pea green color, I guess, and you can see that's more in the stroma lamellae region. And then you have cytochrome B6F kind of all distributed evenly, ATP synthase over by the relatively unstacked stroma lamellae. So that means you have to have mobile carriers that can somehow get electrons coming off of photosystem 2 and get them delivered to photosystem 1. And they're not showing the electron mobile carriers here, but it's just this added complexity. And then of course they've gotten all the way down to the molecular, you know, they've picked apart the proteins and the cofactors. And so if you've taken Intro to Cell and Molecular, this might look familiar to you, the beta, what is it, the beta sheets and alpha helixes and beta sheets. And so this is mostly showing the protein configuration. I just think it's pretty, it looks so cool. So they, you know, they've really gotten into it. They've really are trying to nail this down. All right, I think we're gonna, go ahead.
You say why that like photosystem 2 is in the 1 section?
Why? Good question.
There's got to be some sort of evolutionary pressure for it, right?
Right? But they don't know as far as I can tell. I've looked into it. Every year I look because there's some key things that are still unsolved, and every year I teach this class, I try to see if they've updated, and so far I've been not seeing any updates. So we'll stop here. I will look at the chat. It looks like we had an active chat, so I will see what we got going on there, and I will send out an email to the whole class along with study tips. Some more students were asking about study tips, so I will do that as well.
Someone asked when the exam is.
Yeah, and I'm thinking, you know, because bacteria were the first to do this, you know, and so I'm thinking it's a holdover from that, but I just don't know what the ultimate reason would be. You know, because chlorophyll wants reliving, so I'm thinking it's a holdover.
Compared to